This is a production of Cornell University.
So what I'm going to do today is to give you the talk that I've given many times now in various countries, because at the Department of State there is no plant science department, horticulture department, or anything like that. I was in the Bureau of Economics and Business. So I realized that my home is now on the other side of the Ag Quad. At least I've got to find out what happens over there. But a lot of the people I interacted with were economists and lawyers. But I was in the State Department as a science advisor, and the area they wanted me to work in was agricultural biotechnology. In fact, I was in the Office of Agriculture, Biotechnology, and Textile Trade Affairs within the Bureau of Economics and Business. Our job was to promote American business overseas, but also to promote what they termed food security, because that is a major thrust now of the State Department and the US government, because obviously food security is important to world security, and we don't want to be sending the Department of Defense to every little country throughout the world. So it's really to provide, hopefully, political stability. So this is the sort of dialogue that I've been dealing with all the this past year. So I'm going to go fairly quickly through this talk, simply because you know all the science parts of it, or should do, and you probably know some of the data that I'm going to present as well. So I'm sure some of it will be new to everybody, but a lot of it will be old hat. So if there's some parts you don't follow, we can come back to them at the end. All right, so as I said, try that. No, we'll try this. There we go. So my job was to promote biotech crops. What are biotech crops? They're obviously crops that have their traits modified using modern genetic engineering. And I should point out this talk is also based so that it can be given to people who don't have a scientific background. And as of course, as you know, these traits are determined by DNA. Now, thank you. It is US government policy that we do not use the word GMOs. That is not acceptable. We term these crops biotech crops. And the reason for this is that virtually all our crops are genetically modified by breeding by humans for many hundreds of years, if not thousands of years, which gives us all our modern varieties. And the classic example of this, of course, is modern corn, which looks nothing like its wild grass progenitor, teosinte. And this is entirely the work of human selectors. So many of these statements coming up I'm not going to bother with. But if we compare traditional breeding, I had the pointer somewhere. I'll have to get another one out. It's all right, here comes another one. If we compare traditional breeding, where you're crossing two lines, obviously your new variety is a mixture of the two, and you're going to bring in many characteristics that you don't particularly want. By comparison, genetic engineering does what plant breeders have been doing for many years, but it puts in one desired gene into your variety so that you have only the characteristic that you want and not a whole lot of unwanted characteristics moved around. And there's one thing that most members of the public do not appreciate, and that this is DNA is the same same instruction in all organisms. And one very nice analogy that I heard was that DNA is like a cookbook. And if you have a good cookbook, the chef who reads it can create the same dish. And so DNA is the recipe, and it will do the same thing regardless of the organism that it is in. And so there is nothing inherently wrong with moving a gene from a non-plant organism into a plant. And this gene can be from another plant where crossing is difficult or impossible, or it can be from a completely different organism such as a bacterium. And so at this point, this is where we get to the audience participation part. And I'd like you to all get out your clickers. Oh, wait a minute. We're not in class. You forgot your clickers today. Okay, we're going to do a show of hands and see where we are. I'd like you to tell me how much of your DNA sequence you share with the banana. Would you please raise your hand without looking at the rest of the audience? No cheating. If you have clickers, you can't. You have to. Okay, let's start off with A, 0. No, okay. B, 1%. C, 10%. We have about 3. A D, 50%. We have 30 or 40, I don't know. 90%, about the same as 50%. Boy, some of you are bananas, aren't you? Real bananas. The answer is approximately 50% of your DNA is similar to that of a banana, which normally gives the comment, I'm a banana. This is the sort of thing which is sort of interesting to non-science audiences. You were on the high side. Many of the non-science audiences are somewhat on the low side here. And of course, for those of you who've been around quite a time, may remember the chefs of Manhattan when Flavor Saver Tomato came out. The chefs of Manhattan held a news conference and they said, "We are not going to serve any food that contains DNA." So this gives you the sort of background in which somebody promoting biotech crops to a lay audience has to work. So let's go on. Why are these crops important? Well, let's look at what we're facing at the moment, and this is the increase in the human population, which is currently, as of last fall, 7 billion and predicted to go to 9 billion by 2050. And this means we have to have more food. This is the predicted global food demand over time. And the challenge, the demographers and the people in this field tell us, is to produce enough food in the next 38 years to meet the projected demand by 2050, and it is predicted that we need 70% more food by 2050. And one of the solutions to this is to increase the use of biotech crops. So what are the benefits of these crops? I'm going to go through these pretty quickly because I'm going to be coming back to them. They produce increased yields. They have the incorporation of insect resistance, which decreases the use of toxic pesticides. They have a resistance to crop diseases. New lines are tolerant of drought. Products in the pipeline have attributes for improved health and nutrition. Because of the higher yields, they use less land, so more land is available for nature. Some of the The production methods enable less till, which prevents soil erosion. And also the new lines enable less fertilizer and fuel use, so contributing less to marine pollution and global warming. Here's the latest data on the adoption of these crops. Actually, what is amazing about Cornell is many years ago I found this on the web, so I said, well, where do I manage to download this? So I checked the address. It was Bradfield Hall. So I just emailed the person in Bradfield Hall, and I get the entire slide download. So if anybody wants these, you can get them right next door, though it actually, the organization is based in the Philippines. At least, are they correct, Peter? I think that's their headquarters. I thought he lived in the Caribbean. Yeah, he lives in the Caribbean. Okay, so this is over the years, and as you can see, the adoption, well, this is worldwide, this is developed countries, and this is developing countries. So despite opposition in places like Europe, there is continuing increase in adoption in these crops. And if we look at different crops, we find the highest adoption rate is actually proportionally in cotton. This is the total number of millions of acres. And this is the Biotech is green, dark green, and the total is the full size. So as you can see, the majority of cotton and soybean are Biotech, a good proportion of the maize, and some proportion of the canola. Okay, so for the people who are new to this, very quickly how genetically engineered crops are made. And Cornell, of course, plays a big part in this. And the first is the work of F.C. Stewart in the late 1950s, who was the first to show that you could take the cells of a carrot, a root, and using culture techniques involving various hormones in correct sequence, you could reproduce an adult plant this without going through the flowering stage, simply because individual cells started as behaving as embryos in tissue culture. The agents of transformation, and again, most of you are fully familiar with this, but just to complete the story, is of course, one is agrobacteriums, the crown gall bacterium, and starting in the early 1970s, probably late 60s, when they realized that tumors could be found free of the bacterium, because what had happened is the bacterium had transferred some of its DNA to the plants. And this had become incorporated into the plant's DNA. So the next thing was to remove the tumorous properties which are associated with plant hormones, so remove that, but retain the ability of the bacterium to transfer DNA. So we have the following. Here is an agrobacterium with the plasmid. The plasmid is taken and cut, and also a DNA containing your desired gene is cut. The two are spliced together to make a recombinant plasmid. This is using agrobacterium inserted into the chromosome of a plant, resulting in a new transformed plant. The other one is using the gene gun or microprojectile bombardment, and as most of you know, this is also a Cornell invention, actually up at Geneva. And the idea here is to shoot genes into the plant, which, if you haven't heard the story, this was in about 1988. Most people thought this was totally crazy when it was proposed, but an internal grant body gave Sanford the money, $50,000, to try it out, and to everybody's surprise, it worked. Cornell sold the invention for $5 million to DuPont, and I suspect wish they hadn't. So here's a picture of a technician using the more recent version of the gene gun, which relies on a change in air pressure. I'm not going to explain how it works because I'm sure most of you know it, and if not, those of you who are students are probably going to be taking a course in which this is given. So this enables, by firing gold particles surrounded by DNA into plant cells, the DNA miraculously becomes incorporated into the plant's DNA, so that this can be used to put DNA into plants which are not infected by agrobacterium. So as a quick summary on how biotech crops are made, we have here the method using agrobacterium, and here is the gene gun. And so genes are introduced to the plant using a natural bacterium, agrobacterium, or the gene gun. And then the final step is that these plants have to be regenerated in tissue culture using the methods initially discovered by F.C. Stewart and his colleagues in this building. So let's look at the biotech crop traits that are out there now. First of all, I'd like to cover insect resistance through Bt toxin. So here's the problem. The problem is that there is a corn borer, this little caterpillar here, which eats the corn, causes severe losses. But also, not only does this cause losses, but damage to the corn ears can result in infection, fungal infection, leading to the production of mycotoxins, which are carcinogenic. So there's a double problem with insects damaging corn ears. So what is the answer? The answer was found in the bacterium Bacillus thuringiensis, from which, of course, we get Bt. And Bacillus thuringiensis produces a protein which is lethal to many insects because it inhibits an enzyme in the gut. It reacts actually with the gut wall so that the insects cannot digest the plant material. Now, this horrifies many opponents of, and I'm going to use the word here, GMOs. You're putting an insecticide into crops, but it is standard organic practice that you can spray Bt on your crop and it is organic. And here's a bag of the bacterium, obviously with a lot of clay mixed with it, because you wouldn't have enough volume to work with. But there is the natural bacterium. And so what the scientists have done is put the gene for Bt into various crops so that it contains the Bt protein, which is lethal to any insects that eat it. And this offers season-long protection against insects. And the main advantage of this is twofold. One, to the farmer, that you get insect crops free of insect damage. But also, there's a great reduction in the use of insecticides, some of which are highly toxic. And this, again, is something which many in the general public do not appear to appreciate. And here's a case from, in fact, the Philippines in this. And in the Philippines, the damage to eggplant. Eggplant is a staple vegetable in the Philippines and also in parts of India. The damage is absolutely incredible. I went to an organic farm and there was almost nothing edible on the eggplants. So if you go into a Philippine market, you see beautiful eggplants. How do they do it? Well, they take a sugar cane, cut it across so they have a deep cup, and they put insecticide in there, and they go round, and each fruit is dipped every 2 days in this insecticide solution, and it is the children who often do it with no protection at all. So you've got this, the insecticide level in these fruits is, when you buy them in the market, is probably extremely high, and also we have the damage to the people who are doing the farming. And here is the eggplant, Philippine eggplant. It's called talong in the Philippines, and they have managed to put Bt into this eggplant, and I saw the trials there, and they are completely free of insect damage. And one thing we need to note is that Bt is specific for these Lepidopteran or Coleopteran insects. It does not affect bees. The next one we need to mention is in weed control, and an important character, one of the main characters that's been put into crops, is herbicide resistance. And the principal resistance is to glyphosate. And glyphosate is a much safer herbicide than many of the older herbicides. And it's safe for 2 reasons. It targets a process that is only in plants and not in animals, and also it breaks down rapidly in the soil. So how was this done? Well, to do this, we need to ask what glyphosate does. A glyphosate kills plants by inhibiting an enzyme in a biosynthetic process that leads to amino acids, phenolics, and lignin. And so there's a very short summary of the pathway. So glyphosate acts by inhibiting this pathway. Animals do not possess this pathway, so it has no effect on animals. So what was done is to incorporate glyphosate resistance by adding a single glyphosate-tolerant enzyme from a soil bacterium that does exactly the same synthesis, so getting round the block. And this gives us what is referred to in the industry as Roundup Ready, because the main glyphosate product, glyphosate was originally patented by Monsanto under the name Roundup. And these plants are now unaffected by glyphosate, so the vast majority of our soybeans and some other crops are now resistant to glyphosate. So there's a picture of a US soybean field completely free of weeds. Now, in some countries of the world, these products are available, but only in the simplistic version. And in the United States now, we have what are called stack traits. Stack traits means that these crops have not just one gene that is Biotech, but they have multiple genes. And one of the latest, for example, has 8 genes instead of just 1. 3 Bt genes for resistance to above ground pests, 3 Bt genes for resistance to subsoil pests, and 2 genes for herbicide tolerance. And one thing I need to emphasize from this is Bt is not just one protein, but there are many modifications of it that vary in their effect on different plant pests. So these traits now protect against above ground, below ground pests and give broad herbicide tolerance. And these are now becoming the majority crops throughout the main crop producing areas in states such as Iowa. I'd now like to turn to disease resistance. And for the next few slides, I'd like to credit Sarah Davidson, who took these slides in Hawaii and Thailand. And this is a quick summary. She gives a much longer talk about this. But this was the first incidence of transferring resistance to disease to a crop. It has been pointed out to me that while this was the first time it was done commercially, it was done first by on a theoretical, not theoretical, but on a scientific basis by Roger Beachy, who was also here at Cornell in the 1970s. So the disease in question was papaya ring spot virus. And papaya is a crop in Hawaii, and these are in Hawaii. This is a papaya plantation before the disease and after the disease, so you can see the devastating effect. And the ring spot virus resistance was put into papaya by Dennis Gonsalves up at Geneva. And here's a papaya fruit with the disease. This is the extreme of the disease in the leaves. Going around the Philippines, it was fairly obvious that the majority of papaya had this disease in a mild case. A quick picture of the virus and the leaf hopper that transmits it. And so what Gonsalves and his team did was to transfer a gene for the coat protein to the papaya using the gene gun, and the papaya gained resistance to the papaya ring spot virus. And so here is the difference. This, I believe, is in Thailand. Here's a papaya with the disease. Here's a disease-resistant papaya, and you can see the difference. But then what happened? Greenpeace arrived, and with the theater typical of Greenpeace and the opponents, pretend that this is highly dangerous, and as you can see, this is all for the press, and they destroy the trials. And while I was in the Philippines, there were attempts to destroy the trials of the BT eggplant. This is Sarah's pictures of the protests, for which there is absolutely no scientific basis. And this caused the government to stop the trials and destroy them all. And as you can see, there is extreme opposition to these papaya, except when Sarah walked around to the front of this gentleman, the farmer, we find that, in fact, he is very much in favor of them and trying to point out that if he's going to grow papaya, he wants a fruit yield and not a sick tree. So there are several other crops in the pipeline, particularly crops like banana in Africa, cassava in Africa, for which disease resistance is being incorporated. Some of the other traits, enhanced nutrition, for which the main example is golden rice. Golden rice is now up for approval in the Philippines. It's taken a long time to get there for two reasons. It had to be improved, and secondly, the very extensive trials at the nutritional level and the crop level, which need to be done before approval is granted. And the Philippines has a very rigorous biotech crop approval process. So golden rice is high in the precursor of vitamin A, which prevents blindness, which is prevalent in children in Southeast Asia. And golden rice is high in carotene, which is the vitamin A precursor. And rice plants naturally synthesize carotene in their vegetative tissues, but they don't do so in the grain. So by the addition of two genes with the right promoters, the pathway was reconstituted in the endosperm, so that the endosperm is in fact yellow. And these genes initially came from daffodils, but because of the sensitivity to the fact that daffodils do contain toxins, the work was redone, and so the genes now come in fact from corn. The next series of crops coming onto the market, they're on this year, starting in January, crops that were tolerant of drought came onto the market. And this is thought to be one of the most important developments for countries like Africa, which are facing increased drought. This is a map of the world, and the areas in orange are the areas where it is predicted that there will be less rainfall with climate change. And we're already seeing that, of course, across the Corn Belt of the United States this year. The climate change experts predict that we will be seeing over the next 38 years about 27% less water in many agricultural areas of the world. And this means that it is extremely important to develop drought-tolerant crops. And this has been done, and I'll come on to how it's done in a minute. There are various ways of doing it, but here is, they have drought-tolerant corn. This is drought-tolerant corn. This is normal corn. And so you can see it is surviving where the normal corn is dead. How is this done? It turns out that one of the problems of desiccation is that proteins and other big molecules do not fold correctly. And as you're all aware, protein folding, RNA folding is very important to correct function. And the new drought-tolerant crops that were available for the first time this year, it's corn. They have a protein from a bacterium that the bacterium makes when it receives a cold shock. And in fact, cold and drought can have very similar effects because cold produces desiccation in plant cells. So what the gene transferred is one that enables the correct folding of RNA, and the name for this in biochemistry is a chaperone. Oh, that was the wrong chaperone, so here's the right chaperone. So the RNA folds correctly. Another thing that is extremely important is crops with improved nitrogen use efficiency. The main problem from this is, particularly in American agriculture, the farmers use too much nitrogen because it's all dictated by economics. And this nitrogen runs off down the Mississippi, causing a dead zone at the mouth of the Mississippi. In addition, the excess nitrogen fertilizer leads to emissions of nitrous oxide. And nitrous oxide has 300 times the global warming potential of carbon dioxide. So you don't have to get huge amounts coming off to be a significant contributor to global warming. The genes have been put into plants, particularly they've started with rice, that increase the nitrogen use efficiency. This is high nitrogen use efficiency rice, regular rice, grown on reduced nitrogen. And the reason, the gene is alanine aminotransferase, but the people who did it have apparently they don't really know why it improves nitrogen use efficiency, but it does. The idea here is that nitrogen inputs could be reduced, both for cost purposes in developing countries and also to decrease pollution in developed countries or places where the farmers are able to afford higher levels. What are some other products in the pipeline? There are products with enhanced nutrient content, increased anthocyanins, for example, in tomatoes or oranges, and already available are plants with changed oil characteristics that are better for use in commerce. What are the advantages of these crops? Well, first of all, they increase productivity. This is the increase in productivity due to insect-resistant corn. This is insect-resistant cotton, and these are various countries. And these are the increases in productivity in these various countries following the adoption of these insect-resistant varieties. And the most notable is India for corn, where an improvement of yield of over 50% has been achieved. Let's look at corn in the US. If we look at corn yields with time, and this is bushels per acre, which most people outside the US have no idea what it is. So I have to put it, this is 11.3 metric tons at the top there. Open pollinated. Then in the 1930s, hybrids started and have gained a continual increase in yield. And we now, in 1996, Biotech crops started, and we see the yield continuing to go up. And in 2010 and also 2011, the top test yields were 300 bushels per acre, 19 tons per acre. There's been 11.3 tons, so we're up above the ceiling. This is average yields in Indiana, but this shows the potential that can be achieved with these biotech crops. This is a diagram from plant physiology where there are predictions on the contributions of various techniques to increase yield, corn yields, with years into the future. This is agricultural techniques. The light green is conventional breeding, the dark green is marker-assisted breeding, and the yellow is biotech. And so, as you can see, it is predicted, and this is predictions, but it is predicted that the contribution of biotech to genetic engineering towards yield increases will be even more significant in the future. At the same time, we have a reduction in pesticide use. This is the reduction, this is zero, this is the reduction in pesticides. This is the reduction in herbicides. This is the reduction in insecticides. The blue is the reduction in the amount of chemicals. The red is the reduction in the environmental impact. And as you can see, I don't know if you can see the figures, we have a reduction in insecticide use of almost 80% with the adoption of insect-resistant crops, and the reduction in herbicides in the region of 20%, but because the herbicides are more benign than the older herbicides they are replacing, the environmental impact is about 25% less. There's also a reduced effect on the environment, because the higher yields mean less land is needed for agriculture and can be available for wildlife. In Europe, the European Parliament has told farmers they must set aside 7% more land for wildlife. And so this is one way it can be done, because we can see the following. This is a calculation of the amount of land in millions of hectares, the additional amount of land that would be needed if biotech crops were not used. So in 2009, if biotech crops had not been used, 12 million more hectares would be needed worldwide to grow the crops. Well, this is just a number. What's 12 million hectares? It is approximately the agricultural area in France. So you've got to find an additional France, and France is quite an agricultural country, an additional France to grow the crops if all biotech crops were no longer used. So the last questions we come to for the opponents, are they safe? Well, scientifically, there's no reason that they're not safe. And in fact, they have been extensively examined by US and European science and food safety authorities and declared to be safe in that they are equivalent to non-biotech crops of the same species. They are tested more than any other type of plant production. For example, many of our conventional varieties are produced by radiation, mutation, and then breeding. I was asked to go and find out how many. And actually I was able to come back with this answer within about half an hour. And it is quite amazing how many of our current varieties, including new varieties, have been produced by radiation. Anybody like to suggest a figure that they might, how many of our current crop varieties were radiation induced? Okay, we'll have another show and tell. How about 10? 100? 1,000? Total of 10,000? It's not quite that. It was, I found 3,800 listed. There were radiation-induced varieties, and yet you don't hear any opposition to the use of radiation. And yet radiation, we know much less about what it might produce than genetic engineering. They've been consumed by Americans for over 15 years with no ill effects. In my tours, I've had to deal with Greenpeace at the back. I've spoke to the media, and here's a question I got from the media. You say that they've had no ill effects on Americans, but we all know that America has an obesity epidemic. It's quite clear that these crops are causing people to become obese. So you have to sort of talk about the diet of certain Americans, you know. Oh well, there's not a single documented case of illness or allergy due to the use of these crops, and the environmental effects of these crops are no different from those of conventionally bred crops of the same species. I'm going to skip that. It takes too long to read it. So here are some potential problems. What about superweeds? Well, there are no such things as superweeds. They're just resistant to glyphosate. And if you want to get rid of them, you have to use something other than glyphosate. What about superbugs? There's no such thing as superbugs. They're just resistant to one form of Bt. Both can be prevented by good farming practices, such as rotations of crops and herbicides. And it is recommended to farmers that they interplant a small percent of non-Bt plants with the Bt plants to prevent the rise of insect resistance. And in fact, with these new stacked trait corn varieties, when they are sold, 5% of non-Bt corn is mixed in to make sure that this is followed to prevent the rise of resistant insects. The problems that are faced by farmers and proponents in the industry of for biotech crops is extensive opposition that is not based on science. We have the problem of delayed approvals or asynchronous approvals, where it's improved in one country but not another, so cannot be used for export. It is the European Union has rules that after submission, an approval, if you add up the various stages, is due in about 9 months from submission. However, some are running into 4, 5, 6 years before approval, by which time the technology is totally old and outdated anyhow. So this is a worldwide problem, and this is something the office I was dealing in had to deal with all the time. Another problem is tiny amounts of this in commerce where it is not approved. Using PCR, you can detect these crops even if you have a little dust at the bottom of a shipping container. So, and we've had to deal with, at the State Department, quite a few cases where entire shipments worth tens of millions of dollars are blocked because of a tiny amount of a non-approved biotech event in the crop. In summary, these crops represent innovation, a more precise method of crop improvement, competitiveness for the farmers because they have higher yields with fewer inputs, and sustainability because they have less impact on the environment. Because of the increasing human population, we need these crops to sustain the world ecosystems and feed the increasing human population. And for this, we have to use all the scientific tools at our disposal, and this includes biotech crops.
This has been a production of Cornell University, on the web at cornell.edu.